All right, so section 3.6, metabolism, energy, or metabolism and the energy reactions. So there's a lot to cover in this section. I want you to understand the relationship of products and reactants in metabolic reactions and identify the role of an enzyme in a metabolic reaction. Then you should also be able to summarize the roles of anaerobic and aerobic pathways in energy generation and illustrate the stages of the ATP cycle. So we have a lot to cover in this section. All right, starting off with the metabolic pathways. Reactions normally occur in a sequence. Products of an earlier reaction become the reactants, also known as substrates, of a later reaction. So just to start us off, if we were converting water plus carbon dioxide plus energy equals glucose, the reactants would be the water, the carbon dioxide. That's what goes into the reaction. The glucose is what comes out of the reaction. So reactants are what goes in, products are what comes out. In the case of an enzymatically controlled equation, we call the reactants substrates. So if you see that terminology, that's what it means. It's the reactants of an enzymatically controlled reaction. So such linked reactions form what is known as a metabolic pathway. So it begins with a particular reactant, and then it proceeds through several intermediate stages, and then it terminates with a particular end product. So in other words, the product of the first reaction is going to be the substrate for the second reaction, and so on. So, if we're converting A into B, A is the reactant or the substrate, and B is the product. When we're converting B into C, however, B is now the substrate and C is the product. If we're converting C into D, C is the substrate, D is the product, so on all the way down the line. So here's a metabolic pathway. Enzymes are going to be a very necessary part of the metabolic pathway. Typically, these enzymes are protein molecules that function as catalysts, meaning that they aid in speeding up the chemical reactions. They help facilitate chemical reactions. The reactants, like I said, of an enzymatically controlled reaction are called substrates. The substrates for the first reaction will be converted into products. Those products then serve as the substrates for the next enzyme catalyzed reaction. So that's sort of the metabolic pathway. Now, the thing is that each one of those reactions in a metabolic pathway is going to be accelerated by one enzyme. So if I back up, if this is our metabolic pathway, going from A to B is going to require a very specific enzyme. That one enzyme can only convert A into B. It cannot convert B into C. It cannot convert C into D. This enzyme is going to be responsible for having one job, converting A into B, A into B. And this is how we can actually control the rate of a reaction by increasing or decreasing the presence of that enzyme. So like I said, each enzyme is going to accelerate a very specific reaction. Each reaction in the metabolic pathway is going to require a unique and specific enzyme. And the end product will not be formed unless all enzymes in the pathway are present and functional. In most cases, only one small portion of the enzyme interacts with the substrates. This is what's known as the active site. The active site is where the enzyme is going to bind to the substrate. And when the enzyme binds to the enzyme, the active site will undergo a very slight change in structure. This will allow the substrate to bind more easily. Enzymes are one important thing to note in an enzyme reaction is that enzymes are not used up by the reaction. They simply help speed it up and then go on their merry way. And we'll take a look at that over here. So just as we talked about the synthesis and degradation of macromolecules such as proteins, carbohydrates, nucleic acids, a lot of times this synthesis occurs way too slowly to actually sustain life. So we're going to need enzymes to help speed up this reaction. So what's going to happen is that in the case of a synthesis reaction, here we have a synthesis, we're going to take two substrates. They're going to bind to the active site, that would be this portion right here, this portion of the enzyme. Once they bind to their portions on the active site, that enzyme is going to change shape just slightly. It's going to snap those two pieces together and send them on their way. It's kind of like if you were to sit down and you needed to you needed to bring Lego pieces together. So you have 2 Lego pieces and you want to snap them together. Now, you could either sit there and shake and shake and shake. You could put them all into a container and shake and shake and shake and shake and shake and shake that container as much as you want, if what you want to do is snap those 2 pieces together. Now, that's going to take a lot of energy. You're going to be shaking for a very long time if you want to hope that 2 pieces will suddenly just snap together. It's totally possible. Given enough energy and enough time, you could snap those pieces together. However, what would be much more efficient and much more effective is if you were to take your 2 hands, pick up those 2 pieces, snap them together, and then, set them aside. Once you've done that, you can keep reaching into the bucket. You can keep reaching into the bucket, grabbing two Lego pieces, snapping them together, and then setting them down. Snapping them together and setting them down. You are not going to be used up in this process. Your hands are not going to suddenly stop working after you snap those pieces together. Your hands are not part of those pieces. When you build your Lego house and you've set it down, your hands are not still part of that structure. Your hands are not part of the product. What you're doing is you're taking 2 substrates, snapping them together for a finished product. The enzyme is not part of that reaction. The enzyme is not a substrate. The enzyme is not a product. But the enzyme helps those substrates become the product. Your hands are going to help those 2 Lego pieces snap together and move on their way. So that's a synthesis reaction. The same thing can be said for a degradation reaction. You could take your Lego house, you could put it in a bucket and shake and shake and shake and shake and shake and shake. And eventually those pieces might fall apart. It's totally possible, but it's going to take a lot of time and a lot of energy. What would be more efficient is if you picked up those substrates with your hands and you snapped them apart and send them on their way. And that's how this enzyme is going to work. Those substrates are going to fit into this active site pocket. Once they fit, we'll have an enzyme substrate complex. The enzyme is going to undergo a conformational change where it'll sort of snap apart and it'll separate that initial substrate into 2 products and send them on their way. Just like before, the enzyme, if you use your hands to break apart these Lego pieces, your hands are not still in the Lego bucket. You're not leaving your hands behind. Your hands are not part of the product. Your hands are just involved in speeding up the reaction. So the way that enzymes work is by lowering the activation energy. Molecules frequently do not react with one another unless they are activated in some way. Energy is going to have to be added to at least one reactant in order to initiate the reaction. This is what's known as activation energy or the energy of activation. You'll see it phrased both ways. Enzymes work by lowering the activation energy. They bring those substrates into contact with one another at the right time with the right amount of energy so that way they can snap together. There's still going to be a little bit of energy required. Whether you're sitting there and you're shaking and shaking and shaking that bucket of Legos, that's a lot of energy that's going to be required. Picking 2 pieces of Legos out of a bucket and snapping them together, that still is going to require energy. There still is going to be a little bit of an input of energy in order to get that to happen. However, it's going to require significantly less energy than shaking that bucket. So what happens is that enzymes lower the activation energy. They lower the amount of energy that is required for that reaction to happen, that is required to bring those 2 Lego pieces together. So that's how enzymes work. Enzymes work by lowering the energy of activation, the energy that is needed to start a chemical reaction. Some enzymes are going to be aided by non-protein molecules called coenzymes. Vitamins often play a very important part of coenzymes. This is why vitamins are such an important part of our diet, is because a lot of times they are necessary to activate those enzymes, to activate these reactions and drive them forward. All right, so that's sort of an overview of energy and enzymes. The next thing that we want to talk about is the energy organelles. So we're going to start off with mitochondria. These are going to be found in nearly all eukaryotic cells. All animal, all plant, all fungal cells are going to have mitochondria. They're going to be typically smaller than chloroplasts. Their numbers vary with metabolic activities and energy requirements for the cells. So for example, the liver cells are going to have as many as 1,000 mitochondria packed inside those cells. They're going to contain ribosomes and have their own DNA because they are going to be independent and responsible for their own energy needs. They're ultimately going to be surrounded by a double membrane. The inner membrane surrounds the matrix. The matrix is going to be sort of the jelly-like center on the inside, and is going to be convoluted, meaning that it's folded back and forth to form cristae. So like I said, the matrix is the inner semi-fluid substance. It's going to contain respiratory enzymes that are going to be involved in cellular respiration. They're ultimately going to be responsible for breaking down carbohydrates into ATP. This is where we're going to use up oxygen and release carbon dioxide to turn glucose into ATP. This is going to produce ultimately most of the ATP that's utilized by the cell. It's going to convert that chemical energy stored as glucose into chemical energy stored as ATP, or that adenosine triphosphate. This process, like I said, is called cellular respiration because it uses up oxygen and gives off carbon dioxide. And that's sort of our overview of our mitochondria. One thing I want to point out is the process of cellular respiration occurs on the cristae and in the matrix. It's going to be a multi-step process that's going to require a lot of interaction between this cristae and the matrix. So that's why this cristae is why this inner membrane is folded back and forth to contain all these cristae. It's because we're increasing our surface area to volume ratio. If you remember way on back when we talked about cell membrane, we talked about how if you have the smaller Reese's peanut butter cups, you're increasing your chocolate to peanut butter ratio. Well, imagine if you wanted to increase your chocolate to peanut butter ratio even more. And so instead of, not only do you have the entire outside is coated in chocolate, but what if the inside was coated with chocolate too? So like the outside is chocolate, but also the inside, you have chocolate swirls back and forth and back and forth and back and forth. That would hugely increase your chocolate to peanut butter ratio. And that's what we're doing here is we're increasing our surface area to volume ratio. We're increasing the exposure of the membrane to the matrix because we want to increase that surface area to volume ratio. ATP. So we want to talk just a little bit about ATP. The production of ATP in this cell is cyclical. When energy is needed, ATP is broken down into ADP and P, so adenosine diphosphate and a phosphate group. When energy is obtained from food, it's that energy that we get from food, the energy we get from breaking down glucose is going to be used to glue phosphate back to ADP to produce ATP. So when we need energy, we break ATP into ADP. When we need ATP, or when we get energy from our food and we need to produce ATP, we get that energy from our food and we use it to produce or to glue that phosphate back into that ADP. So all of this is going to be done through the process of cellular respiration. Cellular respiration is the process by which cells acquire energy by breaking down nutrient molecules produced by photosynthesizers. So unlike photosynthesis, cellular respiration is going to use up oxygen and produce carbon dioxide. And this is going to involve the complete breakdown of glucose into carbon dioxide and water. Glucose is a high energy molecule, while carbon dioxide and water are low energy molecules. So we're going to ultimately be releasing energy through the process of breaking glucose down. Energy is going to be released a little at a time in a stepwise fashion, which is going to allow for the efficient production of ATP from those reactions. ATP is going to be very slowly and very gradually produced at each step. If it were to occur all at once, most of our energy would just be lost to the environment in the form of heat. So we're going to do this in a very specific controlled pathway that's going to include glycolysis, a prep reaction, a citric acid cycle, and the electron transport chain. Like I said, this pathway allows for the energy to be released very slowly. We're going to break down each one of these steps in our next video.